neurons have axons, which initiate and propagate action potentials along their length. In doing so, axons help transfer electrical signals from the dendrites and cell body of a neuron towards the axon terminals of the same neuron, which will eventually lead to the release of a neurotransmitter. In this video, we will talk specifically about the axons of neurons, how action potentials propagate along axons, and how the amount of myelination of an axon affects the propagation of electrical activity along the axon. Regarding myelination, we will discuss the concept of saltatory conduction, which helps speed up the conduction velocity of action potentials. So what I've drawn here is an axon. We know that we have many voltage-gated channels that sit within the cell membrane of the axon. And when the neuron is depolarized enough to reach the threshold for an action potential, voltage-gated sodium channels at the start of the axon open, which allow a lot of positively charged sodium ions to move along their concentration gradient from the outside to the inside of the cell, which causes an even stronger depolarization. This strong depolarization kickstarts an action potential, which travels in a wave-like fashion away from the cell body and towards the axon terminals. Specifically, the depolarization at the start of the axon triggers the opening of voltage-gated sodium channels at the next part of the axon, which will cause depolarization here and will therefore trigger the opening of voltage-gated sodium channels at the next part of the axon after that. This process will continue all the way along the axon. Essentially, the opening of voltage-gated sodium channels at adjacent parts of the axon allows the action potential or the wave of depolarization to move along the length of the axon. Yet, the sequential opening of voltage-gated sodium channels along the length of the axon can be very slow and ineffective. For example, when sodium channels are opened, this takes a bit of time, and sometimes when there are many channels open at once, ions and hence charge can leak out of the axon. So, how do we fix these problems? Well, perhaps if we place an insulating cover around the entire length of an axon, aka a big long myelin sheath, this will stop some of the leakage of ions. I have another neuron here with a single myelin sheath that spans most of the axon. In electronics, insulators are materials or objects with a high resistance. So having a high resistance covering, like myelin, around the axon will prevent the leakage of current across the membrane. Because the resistance that ions must move against if they want to leave the cell is too high, the current will want to stay within the neuron, to put things very simply. So, if we leave a tiny bit of axon with sodium channels exposed at the start of the axon, we can have that initial depolarization to kickstart an action potential. And then our big, long insulating myelin cover can hold onto this charge and passively send it all the way down the axon. This will stop the need to open adjacent sodium channels along adjacent parts of the axon, which, although propagates a wave of depolarization along the axon, can be very slow. Because we don't need to open ion channels, but instead we rely on the passive spread of electrical activity through the cytoplasm, we can speed up the electrical signal as it's sent down the axon. In our example, the passive movement of electrical signal through the cytoplasm of the neuron is due to an electrotonic potential initiated at the first part of the axon, which is essentially a movement of charge inside the neuron. independent of ion channels. Electrotonic potentials are very strong at their source, aka at the start of the axon in our example, where the voltage-gated sodium channels cause a strong depolarization. Yet, electrotonic potentials have more decay the further away they are away from their source. So electrotonic potentials don't travel very far distances. Think of an electrotonic potential as ripples in a lake when you throw a rock in. The ripples start off very big, all right, but they get smaller the further away they are from where the rock entered the water. So, by wrapping a single insulating myelin sheath around the axon, although we can speed up action potential conduction velocity by removing the need to open adjacent sodium channels along the length of an axon, which can be a very slow process, the electrotonic potential within the axon will fade out and will be unlikely to reach the axon terminals if we don't have a way to essentially boost the signal somehow. Keep in mind that axons can be very long. In fact, there are some axons of neurons that start all the way up in the motor cortex of our brain and travel all the way down to the lower parts of our spinal cord. So, axons need to send electrical signals over very large distances. So we can't have electrical signals decay in neurons, as electrical signals in axons would never actually result in neurotransmitter release. So, biology has figured out a solution to this. Myelin never wraps around the entire length of an axon, but rather wraps around discrete sections of the axon and leaves some gaps. The gaps between myelin sheaths are called nodes of Ranvier, and nodes of Ranvier have lots of voltage-gated sodium channels. I've drawn a third neuron here with myelin sheaths and nodes of Ranvier. Why are gaps between myelin important? Well, because electrotonic potentials decay the further they get away from their initial source, they need to be boosted. Essentially, each of the nodes of Ranvier provide a part of the axon to boost the electrical signal moving down the axon. Specifically, voltage-gated sodium channels at each of the nodes provide some boosting power for the electrical signal by bringing in some more positive charge, which prevents the electrical signal from essentially fizzling out. So, in practice, what happens in myelinated neurons is that an action potential is initiated at the first non-myelinated part of the axon, and then the electrotonic potential continues this depolarization very rapidly through the first myelinated portion of the axon. The fast electrotonic potential will then cause voltage-gated sodium channels to open at the first gap between myelinated segments, aka a node of Ranvier, which will boost the electrical signal again, and this continues all the way down the axon. Although action potentials do not actually jump from node to node, it may sort of look like this is happening. The movement of nerve impulses along myelinated axons involving electrotonic potentials and the opening of ion channels at the nodes of Ranvier is called saltatory conduction. Saltatory conduction is a reason why we can have very fast conduction velocities of action potentials in neurons, which can sometimes reach speeds of over 120 meters a second in some motor and sensory axons. In addition to speeding up conduction velocities, saltatory conduction increases metabolic efficiency. Specifically, we don't need to maintain the resting membrane potential at the myelinated segments of an axon, which means that we have less sodium-potassium ATPase activity, aka we use less ATP to maintain an electrochemical gradient. Thanks for watching, and I hope this video was useful.